My name is Viking Mayer, and this is my presentation on Drosophila mobility as affected by the Huntington gene. These are the authors. Some background on the Huntington disease is that it is a neurodegenerative disease that affects brain function and mobility along with behavioral issues. This is thought to be due to glutamine repeats or CAG repeats in the first exon of the Huntington gene. There is not much known about the pathways affected by this disease, though we are looking to find Drosophila as a model organism for this disease in terms of testing for behavior and for finding a pathway. Our materials and methods started with genetically modifying Drosophila so that we may cross them and test that progeny. We used a GAL4 UAS system along with the ELAV promoter to only affect neuronal cells, and that those are the only cells we would see expression of the Huntington's gene. There are two Huntington's polyQ genes that we are testing in this experiment, Q16 and Q128, which relate to the amount of CAG repeats we find in the first exon of the Huntington's gene. This is our crosses as we performed in the experiment. On the left, we have Q16 crosses. The only ones that are expressing are in the middle, which are female straight-winged flies. Female curly-winged flies have no expression as they hold a balancer gene, and male flies do not hold the ELAV GAL4 to have expression of the Huntington's gene, while Q128 expression is only in the female flies. We only tested the progeny because those are the only flies that have expression, and what we did was use a reverse gravitaxis in which we tested at the 5-second and 15-second marks how far flies had gone up a graduated cylinder. These are some of the results that we have, in which you can clearly see the Q128 females are heavily between 0 and 1.5 inches, while the rest show a normal or bell curve. At 15 seconds, we can still see Q128 females are under 3 inches, the majority of them, while the other groups, Q16 and Q128 males notably, are at mostly 4.5 inches and above. Q16 females still have a few in the 0 to 1.5 inch range, but they are still more normalized than the Q128 females, which are heavily skewed. So some notable averages when discussing the results are that in the first 5 seconds, Q128 females did not go above 1 inch, and in the 15-second mark, the Q128 females did not go above 1.5 inches, whereas the Q16 females went above an inch in the first 5 seconds, and at about 1.5 inches for the second 15-second mark. What we can clearly see in our results is that the amount of CAG repeats as expressed in the Huntington's gene affects the mobility of the flies heavily. Q128 female flies showed less movement and lower numbers as compared to Q16 and the controls of the males, while we also hope this can be further implemented in higher level organisms like mice, or even to use this Drosophila as a standard for pathway study We also hope that our implications here can be a jumping point for further research in as using Drosophila as a model organism to base behavioral studies off of.